it's these ones. And because of Brexit, yeah, it's 2 months, but more money for each month. So it's a zero-sum game. But effect makes no difference. Just helps a little bit. But it had to be this year because it's a 3-year grant.
Can you make this disappear on your computer?
COVID interrupt us.
Yeah, just continue. Correct.
So is that not focused? Now it's better now. Well, I have even explicit duties from be part of the group and run a little workshop in a couple of weeks' time. Yeah, in 3 weeks' time.
Hello, everyone. I think we can start. I'm sorry about the pizza. Pizza is on the way somewhere in the town, stuck in the traffic. So it will be arriving soon, and we can just grab it during the time. And in the meantime, I think we can progress with the seminar as such. And pizza will just show up. I hope it will be better next time. And so today we have here Ezgi, who is the postdoc at the Martin Wolf's laboratory. She will just tell us what she was working on before when she came to here, right? Yes. Yes. And next week we will have the seminar given again by Roger. It should be more like a round table discussion on the topic, which is called "Where does your research fit?" So we will be discussing the wider things. I will try to be online, but I have to say that quite few people will be in Montpellier. So I will ask someone to set it and we can be online and just sit around. There should be again a pizza available. And the following week, on the 15th of June, there will be another discussion, mostly for PhD students maybe, or younger people, which is called "Should I Get Involved?" which Roger says the social issues as the scientists and how to confront political and ethical social questions within the research. So like that. And then I think we might have a seminar which will be kind of colliding with the workshop for which Roger is here. So then we from the 20th till 22nd of June, there will be the successional workshop, which will be led by Roger. Vojta will be back for it also. But on the 22nd, we should have the seminar by Sarah Segar, also very short one, but it's compulsory because she didn't present last time on the PhD conference. So we will have to do it somehow, spatially separately.
The workshop should finish at midday.
Okay. Perfect.
On the Wednesday.
On Wednesday, once we finish the workshop, Sarah will present shortly because she tried to avoid her duty and she was given just a few days extra to catch up. So welcome everyone today and enjoy the talk. Thank you. So yeah, when the pizza comes, I won't be offended if you come and grab. I might actually do the same. So apologies for that in advance. So welcome, thanks for coming. I am Ezgi. I just started my postdoc with Martin Wolf about 4 weeks ago, so I'm pretty new. And today I'm going to talk about what I did in my previous postdoc in Switzerland, which is floral color evolution and Gesneriaceae, and how different pollination systems have contributed to the diversity in this family.
So just to give like a very brief introduction to like what I will be doing here, like why I'm here before going into the previous project, I'm working on the Gesneriaceae phylogenomics and the evolution of chemical defenses at Martin's lab. And my general like broad interests are phylogenetics. I happen to study plants. I have happened to study plants for now. I'm not very picky. Macroevolution and trait evolution, diversification events. And I usually like often get involved in like a lot of bioinformatic analyses. So that's also like slowly becoming my interest as well. And since I'm not going to talk about my project here now, but I'm going to present a poster in ESAD conference in Prague in August. So if anyone is present, please come.
Maybe let's finish your seminar first and then we can.
Okay. So back to my project or previous project. So the results I'm presenting here, they're already published and they're mostly from this paper. If you're interested, I can send you the link or you can just Google it. Let's start with the plant-pollinated interactions. I find this very interesting, and I think I'm at it's like a proper department, it's like an entomology department, because most of the pollinators are like insects, but also a lot of other animals. And floral color from plants' perspective, it's an form of advertisement to like tell the pollinators that like I have food. So it's it has a very important function in terms of plant-pollinator interactions. And as you may know, there are pollination syndromes. So it's like almost everything in biology. It's like a very overly like oversimplified of the reality, but there's these like general trends of like a floral color or morphology that certain groups of pollinators are attracted to or use as cues to like search for food. So the preference of these pollinators are obviously based on their sensory abilities. So they're attracted to what they can see. Like that's the first like line of differentiation, I guess. So if they can see it first, then they might go to it. So for bee vision, there are 3 types of color receptors in bees: UV, blue, and green, meaning that their color perception ranges from UV, which we cannot see, unfortunately, but it extends only to yellow-ish hues. So they're really not good at discriminating orange or like red hues. This is the main difference between bee and bird vision. So bird vision has, instead of 3, has 4 types of color receptors. So they have 1 extra receptor, which enables them to see the orange-red hues. So they expand the spectrum of their visual capabilities. So orange-red is again like the main difference between these visual systems. And this is not trivial because red colors, like red flowers, are like pretty common. They have evolved in several plant families throughout the evolution, and they're frequently associated with bird pollination. So like one of the main features of bird pollination syndrome is red color, red and orange color. So this is an important thing. Again, like this is also like a main difference between bird and bee visual systems. Going back to the chemistry of floral color, these are like the floral color is given by these pigments that absorb and reflect like selectively certain wavelengths. That's what makes them like different color. And in plants, there are 3 main pigment groups. One of them is carotenoids. They're like very common, very frequently observed in many angiosperm families. They have a narrow color range, meaning that they go from like yellow to orange and red hues that you see. And flavonoids, that has the widest color range in terms of the pigment groups. They can go from anywhere from like yellow to all the way to blues that you see in some flowers. And it is also like very frequently observed in many angiosperm families. And there is also betalains. This is the like a flavonoid equivalent. So this again has a wide color range from yellow to like purple. But the thing is, it's only limited to Calyophyllae, so like cactus, beet, carnation, bees. So it has a very limited distribution in plant phylogeny. And it's never observed with flavonoids. So if you're seeing flavonoids in a flower, it means that there's not going to be betalains and vice versa. And anthocyanins are part of the flavonoids. They are the most widely studied pigments that I'm going to talk about for the rest of this talk. I don't want to intimidate people, but this is the anthocyanin pathway. It's not that complicated compared to other pathways. So it starts with, can I use this? I can use this one or this one. There. So like it starts simple, has a very like common building blocks, and then it branches out depending on the hydroxylation number of hydroxyl groups, and it produces many different pigments, anthocyanidins, depending on the hydroxylation level. So pelargonidin, cyanidin, dalphinidin, these are like very common ones. And again, we also have a lot of modifications that gives even like more like diversity of pigments. So peonidin and malidin here, it's like a metal transferase. So you're adding a metal group, and there you go, you have another pigment that gives another color to flowers. So the color spectrum here is kind of representing the pigment. So pelargonidin is more like orange-red colors, cyanidin, peonidin, dalphinidin, malidin is more purple to the blue shades. And there is a secret alternative pathway in the anthocyanin pathway that this is producing 3-deoxyanthocyanidins. So the main difference is that they're using this hydroxyl group here, and they're diverging from the, branching out from the anthocyanin pathway very early, and then they're producing a different set of pigments that are limited to only orange and red coloration. So I guess you're seeing where I'm going with this. There are different ways of making red color using the anthocyanin pathway. So red flowers. So here are a few recipes. Hydroxyanthocyanins, the regular anthocyanins, the main pathway. Pelargonidin is a common pigment that produces orange-red hues, but it's not the only one, because the others, even though I said they mostly produce purple and like blue colors, depending on the pH or like different like presence of metal ions, or even like the presence of other pigments in the flower, they can produce red as well. And again, the deoxyanthocyanin, the alternative pathway, is producing red pigments, red-orange pigments. Luteolidin, apigenidin are the most common ones. Again, this is a very, very like limited like pigment group. They're not present in like many families. They're only found in ferns and mosses, some Poaceae, in tea, in Theaceae, Bignoniaceae. So there are like few neural species that produce these deoxyanthocyanins. And Gesneriaceae. So the New World Gesneriaceae is, it has, I think, the widest distribution of deoxyanthocyanins. A lot of species produce these pigments. So this is actually like why we're interested in like studying this like, red coloration in this family. So as you can see here, Gesneriaceae is the monophyletic clade, like the subfamily of the family Gesneriaceae. They have about 75 genera, over 1,200 species, and you can see the morphology and color diversity. So like if you imagine a color and like morphology combination, it probably exists in this weird family. They're really beautiful. I like them. So this is a really nice family to study floral color evolution. The same thing with pollination. They have many pollination modes. The three main functional groups are bees, hummingbirds, and some bat pollination. There are so many switches from bee to hummingbird pollination in this clade. So the ancestral state is bee pollination. The old world subfamily is obviously ancestral bee pollination, but hummingbird pollination have arisen over 30 times. Like there are so many evolutionary switches, and these switches also happen back and forth, which is actually rare. Like it's if you're familiar with the literature, hummingbird pollination is usually like a final character that not many switches back to ancestral pollination. This is not the case in Gesneriaceae. So linking all these together, now we have these hydroxyanthocyanins and deoxyanthocyanins producing like floral color in Gesneriaceae. And hydroxyanthocyanins having a wider range of floral colors, they are attracting bees and hummingbirds because of their visual abilities, whereas the deoxyanthocyanins with the limited orange-red hues, they are attracting hummingbirds, like they're more hummingbird pollination oriented. So we hypothesize that plants with deoxyanthocyanin, the alternative pathway, have an alternative way to produce orange-red flowers as an addition to the hydroxyanthocyanins. And this results in reduced like floral color diversity, but a higher frequency of hummingbird pollination. And we're interested if these are linked on like how they are evolving in the family. So the materials and methods sort of, so floral color and chemistry. First of all, we measured the physical aspect of the floral color. So we measured reflectance using a spectrometer. So you can see the color profile, reflectance profile for each species that we sampled, and you can see the significant, like the signifying reflectance curve for each flowers. And then the chemistry, we used HPLC and mass spectrometry to identify the pigments, like what pigments are producing these colors in our subfamily. So we detected these pigments, which I'm not going to go through, so I'm going to simplify it a little bit. So all the pigments I showed on the pathway, we have observed and detected in the samples. We also have detected 2 deoxyanthocyanins. So here is the summary. Overall, like 5 main hydroxyanthocyanins and 2 deoxyanthocyanins that we have detected in Gesneriaceae. So the pigment categories, because we were doing like ancestral reconstruction and like simplifying things, we bin the flowers that has more than 90% of deoxyanthocyanin pigments as one group. Deoxy and hydroxyanthocyanins, they have a combination of these pigment groups, or like hydroxyanthocyanin only and no pigments. So these are usually white or like yellow flowers. And the distribution is, there's a lot of hydroxyanthocyanin dominant species, but also a lot of like a little bit of everything. And so looking at the color diversity, so this is what we perceive, like, so like you can see all the like the visible spectrum, but this is human perception. So we have to, I'm going to talk about this later, we have to see this through the pollinator's point of view, but this is for now like what we are seeing. Color categories is, so again, like to like objectively categorize color instead of just saying like orange, red, or like, because this can be like very subjective. We follow the methods, like a model based on like a classic paper. So in this model, you divide the spectrum into 4 sections, and like UV, blue, green, and red, like 4 main domains. And if you see a reflectance, it means it's positive, so it's there. So it's reflecting in the red spectrum, it's positive. And the other tricky one is sometimes the reflectance is just at the end. So this is coded as like this slash in this like model system. So the, what's happening? No. So this is a distribution of the samples that we used in the study. So these 2 are what we call as like what humans perceive as orange-red. And there is, as you can see, also the pigment types. You see that the deoxyanthocyanins are very like heavily represented in orange-red flowers, as we expect, but there's also a lot of like a combination of both pigments or hydroxyanthocyanin only. So you can already see that the red colors are being produced by a variety of pigment combinations. Again, the other thing to note is the no pigment flowers are usually white or some yellow, which we think is carotenoids that we didn't study. The other thing to note is, so we cannot see UV. So UV plus, minus, or slash, like for us, it makes no difference. Like, for instance, so here and here, and yeah, it's here as well. Like they're all white to us, but they see like pollinators, like bees and hummingbirds, since they can see UV, they look different to them. That's why we categorize them using this model to be more pollinator-oriented categories. Antocyanin diversity in this family, it was surprising to us too, because it's very, very rich. You can see the combinations here. So this is a heat map. So the darker the squares, the more frequent the combination is occur. So this is a bit unusual because in many studies that they studied the same thing in different families, like Solanaceae is a family that this study has been done very like recently. They usually observe like maybe usually one type of pigment, but sometimes 2, combination of 2. But here in Gesneriaceae, we have observed up to 5 different pigments in a single flower. So there's no pigments or 1, 2, 3, 4, up to 5 different pigments happening, like occurring in the same flower, and end result is like some like really crazy floral color diversity. So the PCA plot here is showing the distribution of the samples, and it clusters based on color. So you can see the red ones are here. So PC1 is separating the samples based on red, orange versus more like purple, bluish, like pink hues. And then PC2 is sort of, it's like deoxy groups are clustering together. Like the samples with deoxyanthocyanins are clustering at a different spot than the pelargonidin, so hydroxyanthocyanin one. So you really can see the distinction of different colors and pigment producing, like pigment groups. The phylogenetic distribution of the deoxyanthocyanins, or the alternative pathway, you can see that it has arisen like several times in the family. So you see, like, you don't need to read this, obviously, I can barely read it, but what it's showing here is there are like basically 3 main groups. So this is, should be Nematanthus, yes, Nematanthus genus. It's very rich in deoxyanthocyanins, and they are mostly producing this dark green, so epigenidin, deoxyanthocyanin. There is the glossoloma. It's again like very, there's a lot of like orange-red flowers in this genus producing deoxyanthocyanins, but it's a combination of, so again, like epigenidin, but also another other hydroxyanthocyanidins. And then there is Sinengia. This is one of the largest genera in the family. It's very diverse. And in this one, we see luteolidin. So it's another deoxyanthocyanin, very commonly produced in this genus. So you can see that this deoxyanthocyanin pathway usage has come up several times in the evolution history of the family. So one thing that you may ask, like I did, like first thing I saw this project was, okay, there are two different pigment groups. Can we tell them apart just by looking at them? The answer is no. So looking at the deoxy pigmented flowers and hydrox pigmented flowers, they look identical. And when you look at the reflectance curve, they really are identical. The only difference is the pigments they're producing, but the outcome is the same. You cannot tell the difference. So there really is like the same color, but different pigments, like two different solutions to the same problem, basically. So to sum up so far, Gesneriaceae is like very diverse in terms of floral color and like anthocyanin pigment production. And red flowers can be produced by two branches of the pathway, and both red flowers and deoxyanthocyanin production has like evolved several times in Gesneriaceae. So we're adding pollinators into the picture. As I mentioned, that we're going to talk about this later, the visual systems of pollinators. So we want to see, like, well, try to see how pollinators are seeing these things, like seeing these flowers. So we use visual systems, and when we map all these samples on a bee visual system, you see this. What it means is, so the center of this hexagon plot is the achromatic center. So closer the samples are to the center means like bees cannot differentiate them from the green background. So they're kind of like blending in with the background. And by looking at the color coding, the deoxyanthocyanin producing ones are closer to the center, whereas the others are more like further away from the achromatic center, which makes sense because red-orange color is, bees cannot tell them. For them, they look just like the green background. But in hummingbird visual system, so if you notice, this is like a 2D plot because there are 3 receptors. Hummingbird is more like a 3D, so it's like a pyramid. Because of the fourth, like an extra receptor, but the achromatic center is here, but it's a 3D, so like it's hard to see like what's happening. But just to compare, they have a wider range of visual discrimination abilities compared to bees. To quantify this, we have this concept of marker points. So in the reflectance curve, middle point of a steep curve, so it's like here, this is where the maximum color recognition happens. So this is the significant point of this reflectance curve. And there can be multiple. So this is like a classic red reflectance curve, and this is for purple. So there is 2 and actually like a third one here. So these marker points are important because that's where the recognition is at maximum for like whatever is observing. And when we overlap this with color sensitivity of pollinators or like whatever visual system we're interested in, the better match with their color sensitivity, the better ability to perceive and like discriminate the different like small differences in hues. So what you see here is the red flowers are outside the B visual optima because that's where the peaks are, but the marker point is not falling in these high sensitivity regions. Whereas the purple flowers, which is like a classic bee pollination syndrome, they're like falling right into the high peaks of sensitivity of bees. So let's go a bit faster here. So comparing the deoxyanthocyanins and hydroxyanthocyanins, you map all the samples. I have this like crazy overlap of reflectance curves. So these are the orange-red flowers that are producing the deoxy pigments, and these are the hydroxy ones. As you can see, the color diversity is much wider in the hydroxy ones. And if you look at the, again, like bee versus hummingbird discrimination optima, the peaks, the blue peaks that I showed in the previous one, I just like represented them by arrows to simplify it. The main difference between deoxy and hydroxy is the bee discrimination optima is that we're very well represented in hydroxy plants, whereas the deoxy plants, they are not being perceived well by bees, whereas hummingbirds, it doesn't matter what the pigment is, they really can perceive the whole spectrum. So again, like quantifying, or scientists, we want numbers like p-values, etc. So this is another like model just to quantify like how well they can see discriminate colors. We use mean absolute deviation of inflection points from the visual optima. It sounds super complex, but what it's basically telling is, so this is the marker point, the middle part of the very steep curve. And the MAD value is basically how far this marker point is from a visual optima of a visual system. So for hummingbirds, they can really see well, discriminate the hues well at around 600 nanometer wavelengths. And the marker point is at 605. So the MAD value is 5, and lower the value, meaning that it's a better fit to the visual system. So the lower the value, the better the floral color or any color is perceived by the observer. And when you compare assess the same point, like the same floral color, the bee visual system, their closest optimal point is at 500 nanometers. It is very far, so it's 105. It's much further away, meaning that they have very hard time being able to perceive this color. Yeah, so like I said, low values, the lower the value, the higher the discrimination ability. So in this example, the red color is much better perceived in bird visual system than the bees. Again, the same thing, just using a different approach. We have shown that comparing the bee versus hummingbird systems, the main difference is again the deoxy anthocyanin. The bee values are much higher. They are not able to discriminate well, whereas the birds are doing really well. They have very low values. Okay, so this is just a summary. This is coming to an end, so we can have pizza soon. Floral color in Gesneriaceae, it correlates with the color discrimination, ability of pollinators. So this color system has evolved, got impacted by the visual abilities of pollinators. And deoxy flowers are better match the hummingbird visual system, and hydroxy flowers match both visual systems because of the colors they are able to produce. And the alternative pathway pigment producing lineages are evolutionary constrained because of the floral color spectrum. Because of this, they can only produce red-orange colors. So if you evolve to produce deoxyanthocyanin pigment only, you will only be perceived and potentially, like very probably, visited by hummingbirds only. But is there an evolutionary dead end? Like going from the hydroxyanthocyanin, like a wider range of colors, like producing like whatever you want basically, to like limiting yourself, to limiting hummingbird pollination only? It is not an evolutionary dead end. It actually is an advantageous strategy in Gesneriaceae, at least, because previous studies have shown that the hummingbird pollination has actually increased the speciation events in this family. So hummingbird pollination, like switching to hummingbird pollination, is very advantageous. So, and again, it's not a dead end because, like I said earlier, you can always switch back to bee pollination. So this is basically the same pathway, switching back and forth. It's not impossible. It's actually like pretty easy in terms of genetic function. And like, again, like hummingbirds are very effective pollinators in the neotropics. So like going from bee to hummingbird, it's actually good because in some habitats, they're actually better performance. Hummingbirds perform better in terms of pollination. And again, like it promotes diversification of the family. And to sum up, again, the last point is the floral color diversity in Gesneriaceae, Gesnerioideae, is linked to the use of two alternative pathways. So they're using the anthocyanin pathway up to its full potential. They're combining like whatever is available there, and they're using it unlike any other system, and which provides alternative solutions to the same problem, like I discussed in this red-orange floral color evolution. And it's not an evolutionary dead end, it's actually promoting diversification. I'm going to end my talk here. Thank you for listening. And I would like to acknowledge my supervisor, obviously, first of all, Mathieu Perret in Botanical Garden of Geneva. This project was also part of our master's student carrying master thesis. And we have some collaborators in Switzerland, Colombia, Brazil, Switzerland again, and some funding. And again, one more time, thank you for listening. Thank you. Some questions here?
Very nice talk, especially very interesting use of these animal perception systems to understand in more detail what's going on. I was just wondering if you have any data on the actual visitation of these flowers. If for example, the flowers that are preferred by bees according to the visual system are actually visited only by bees and vice versa?
Yes, not all pollination data are observed. Some of them are inferred, but like in the paper, actually, there is a list that you can see which samples are like observed in the field, in the wild, which are inferred. Mm-hmm. Yeah, we have some data, not all, unfortunately.
But it matches. Like...
It matches, yes. Cool. Yeah. Yeah.
So you have shown in one of the introductory figures that these plants can switch between bird and bee pollination, right? So does it also mean that they are switching between the pathways or between the pigments discretely?
That we haven't checked, like if they're happening as a consequence, or like which comes first, but. I'm guessing like switching is probably happening, like the back and forth switching is happening only with the ones that are producing hydroxyanthocyanins, because if you're producing only deoxy, then like bees do not have much chance of like coming back to life, you know, like into the picture. But yeah, the timeline of that we haven't tested.
If I can, why is the speciation faster or like better when they are pollinated by the hummingbirds? So you said that when the pollination is happening through the birds, the speciation is faster there or better? They diversify more?
Yes, the diversification rates are higher in hummingbird pollinated.
Why is it so?
I think it is because it's like a limiting niche that they're avoiding competition. So this is one theory that like switching to hummingbird pollination syndrome, which is often like red colors, it's disabling bees to like steal resources. So once you go to hummingbird pollination, you found your pollinating system, and you're limited, like you have higher chance of being a specialist. Does that make sense?
I think one point that is quite important is that bees eat the pollen and hummingbirds don't, right?
Yeah.
So I think that influences quite a lot the trade-off in being just a vector of pollen versus transporting some pollen that you didn't manage to eat.
Yeah. So it's mostly like avoiding bee visitation, like going to hummingbird. Like it's not I don't think this is a preference. I don't think hummingbirds are preferring red color because they like the color, because there's always like more food there because bees are not stealing it. Yes.
Please, why is the inflection point of the reflectance curve important and not the maximum? Even if there is the inflection point is only 50% of the intensity.
It's not the intensity, like how far it goes up in the reflectance curve, but the wavelength. So that's the point that I think, like, I don't know the deep physics of it, but like here, so you're looking at the wavelength, and reflectance, like the height of the curve, just means that the hue is brighter or like more pale.
And I would expect that where it is the most bright at the 650 or 700, it would be much better than when the reflectance is only 30% at 600 exactly.
I think the way to explain this is, so
the one with the white flower, I think it's the easiest way to explain
it. Like the very beginning, maybe. What was
Sorry. There.
Here? Yes.
Yeah.
So
yeah.
Here, like how I read this reflectance curves are, so the white one follow the gray one. It's reflecting everything starting at 400 nanometer. So like it's reflecting everything. That's why we see it as white. So the important point is this area, like where there is a sharp change.
Why?
Because this is the key point of like key wavelength. The way the color is produced.
It's just that they can differentiate if it's in the middle. They can differentiate between different shades of the same color.
Yeah, like if there is a peak, if there is a change there in the beginning and nowhere else, it means that the flower has to be white, because it means that everything is bright. It's reflecting in every wavelength. While if it's going up only in a bigger wavelength, it has to be red because it's low everywhere else.
Sure, it's clear, but if
so, what matters for the color is where the reflectance is increasing.
no, it matters everything, and where is the most reflectance, most percentage, is this what you perceive best?
I think this might also explain better, like here, like these color categories. So wherever there's like a sharp increase, it produces that, like it reflects that color. So you see the color. So here it's not, there is no increase anywhere. So like UV, it's not reflecting UV, it's not UV. Blue is the same, green is the same. Like as soon as it comes to red area, so like 600 to 700, there is an increase, meaning that it's reflecting that color very strongly.
Only half the intensity. Not very strongly.
It's the average, like it's this whole area. So it makes sense to take the middle point because the whole area is like contributing to the color.
Yeah, and maybe if you return the previous figure there, because the one flower. No, yeah,
This one.
here you can see, actually, if you would look at the maximum for the red color, you would be able to distinguish it from the white color, right? You need to check where the peak in the reflectance is starting, just to see the difference between red and white.
Yeah, so for red, it's only going up. This like point is around 600. So the red is there, but nothing, no other colors are there. Whereas for white, the like starting like UV, like blue, green, like everything is there. So the end color is like white. It's a combination of blue, green, red, yellow, like everything. Like I hope that helps.
Sorry, I'm not convinced.
I actually have a follow-up question of that other graph which was showing that the line or like no, you could show it. There was a line at 10%. Is it some? Yeah, this one.
Yes.
Is it some kind of already decided the threshold that will?
Yeah, that's not like an arbitrary cutoff
This is
value.
arbitrary by you or by a bird or by bees or
By this model.
by this
These
model?
are the color masters of pollination biology. So they decided to use this model and use this threshold. I just followed their advice.
Okay, but
Because like 10% reflectance is like so low that it doesn't contribute to the actual like outcome, like the color.
okay, nice question.
But again, yeah, arbitrary. Why not 12? Huh?
Yeah.
Yes.
Could be the colors also connected with other floral traits, such as like nectar production and stuff? Have anybody looked at it?
Yes, potentially, like we haven't tested it, but there are studies linking nectar production, nectar content, or even floral morphology. So like the red, like the bird pollinated flowers, the typical syndrome is red, but also like tubular, tube-shaped flowers, so the bird bee can get into, and it avoids also bees, so they don't crawl and like steal their food.
I meant rather the family, like how does it, whether like someone looked at it.
In this family, yes, they looked at nectar. There is like a distinctive bird preferred versus bee preferred nectar. It's the content, different like amino acids concentration or different types of sugars. It correlates with pollination syndromes. Also, the floral morphology is also like a very, like it correlates well. And in fact, the inferences that are made, like the ones that we did not observe in the field, we inferred like bird pollination or bee pollination based on, I think 27, like up to 30 different morphological characters. Color was not one of them, so it's not just like circular like inference. And yeah, this model actually is like matching the observations that are made in the field, so it's a pretty strong one.
So also those groups which switched kind of back, for example, to bees, they again decreased production of some stuff and they switched the shape of the
Yes,
flowers.
probably. There's still like so much fun to do, but
Okay, maybe I will like stop the discussion now. We can leave it for the pizza because there seems to be actually many questions. I will only ask people online if they want to ask something. People online, any questions? Hello. Seems that no. Okay, so let's have the pizza and we can continue the discussion about the colors for now. Thank you.
Almost finished.